Okay, thanks very much for the introduction and good morning to everyone. My name is Ewan McShane and I'm a PhD student from Harriet Watt University in Edinburgh. And here I'm going to be outlining the method for locating fiber optic medical devices deep in tissue using time-resolved imaging. So the method I'm going to be presenting today basically contains three main elements, and that are that we're applying a time-resolved imager. We provide a method of emitting in vivo illumination, and we perform diffuse optical imaging of light emission emitted from in vivo light sources to gain some spatial information on the location of those sources of light. And these three things in tandem really represent a method of locating devices using diffuse light. And the particular application that we're applying this to is locating in vivo medical devices. And the reason why we chose this particular application is that we believe that current clinical methods of verifying the placement of many medical devices are flawed, and that's been clinically expressed clinically. And we believe that we have an optical method that could be employed to verify that placement instead. And we believe that such a technique could change workflows and ultimately improve patient care down the line. So just briefly, before we get into the sort of the meat of the talk, the imaging hardware we're applying here are single photon avalanche diode detectors, that some of you might be familiar with, or SPADs. And we're in the imaging system that I developed, we're actually applying a linear SPAD array in a scanning configuration. So the actual sensor that we're using here was developed by the Henderson Group at the University of Edinburgh. So shout out to those guys for the hardware development. But critically, what a linear array in a scanning configuration like this allows us to do is perform wide field two-dimensional imaging with time-resolved imaging capabilities. But critically, we obtain images that have high pixel density. So in this case, a 2.26 megapixel image with fine temporal resolution on each pixel. And we'll see why in future slides why that temporal resolution is so critical in the techniques that I'm going to be discussing today. So just to briefly sort of demonstrate the temporal and spatial resolution achieved using the scanning imaging system, we perform some basic light and flight imaging. This is just some niceties, but if anyone else is introduced in alternate applications of the imaging system that I'm discussing here, feel free to give me a shout offline or whatever. But what we're really interested in here is imaging photon transit through scattering media. So I'm not going to go into too much detail on this within the realm of this talk. Again, if you want to discuss this more offline, we can do so. But essentially, light emitted from in vivo medical devices, which are emitting pulse near infrared light, a small amount of that's observed to emerge from the surface of the patient or the scattered media. And when imaged in a time-resolved fashion, this light seems to emerge with a large variation temporally, and that's a function of varying path length. The photons are taken from the light source to the surface of the media. And what we observe is that at later times, we see a larger region of illumination as the more diffuse photons have migrated more slowly to the surface of the patient. And what our time-resolved sensor or detector technology allows us to do is essentially time-gate out or discriminate against those later arriving photons. So we can kind of focus on the photons which have taken a more direct path from source to detector. If you want more information on that particular kind of mechanism, you can see a paper by one of my supervisors in Biomedical Optics Express. So moving on, applying this kind of rationale then to diffuse light imaging of otherwise obscure medical devices, we chose to apply actually a bespoke fiber optic probe that we manufactured in-house. And this essentially allows us to provide discrete illumination at many locations along the device line. Three of those are highlighted in this left-hand side image, but the fiber that we applied in this measurement actually had 18 discrete locations that we can illuminate individually. So if you want information on the actual manufacturing process of that, you can see one of our papers is also in Optics Express. So yeah, we added some essentially milk to otherwise obscure the path of the fiber optic device, and then we imaged the surface of that phantom in time-resolved fashion using the imaging system that I previously introduced. And the light emission then that we observed from a single one of our emission points then expanded temporally is shown here. So the earliest time within the measurement window was seen in subfigure B, and time is increasing through the subfigures. And what we observed then is a smaller, more localized spot of light at early times, which seemed to grow at later times as the more diffuse light has migrated towards the surface of the phantom. And this really indicates that this early light may be able to better constrain the actual spatial location of the light source than the later arriving light. And we're using that approximation then expanded across all the light source locations. So just briefly, if you look at the full width half maximum of the light spot in the image at each time location, you see that increases with time. So expanding this rationale then to all the light sources along the fiber optic device, we effectively track many locations along the fiber optic device. And since they're located incrementally along the fiber, we effectively track the entire fiber location over an extended region. So in the absence of scattering particles in the phantom, we gained the known locations for all the light sources. And then using the early photon processing rationale, we extracted some recovered locations, and these two were seen to correlate quite well with each other. But moving on to the real interesting application, let's say, of what we're trying to do here, and that's locating in vivo medical devices. So within pre-existing medical devices, such as interior feeding tubes, we place fiber optic probes, and from those we're emitting pulse near infrared light in the optical window. So basically, if you don't know what that is, like light in this wavelength range can penetrate through larger portions of human tissue with respect to other wavelengths. And we image the surface of the patient in the same fashion as we've seen previously. So what we did was we packed up our imaging system, which was a bit of work in itself. So that's the actual imaging system suspended above the patient that you've seen in the initial slide or the second slide. And we used a porcine cadaver model to test this. So we had a clinician essentially place a fiber optic probe within the stomach of the pig, and then we imaged the light emission then on the surface of the patient. Before doing that, we verified the location, the known, let's say, our ground truth spatial location of the light source using CT imaging. We then compared that and aligned that to our time-correlated imaging. So within this subfigure, the circle there really denotes the expected spatial location of the fiber optic probe within the stomach of the pig. And then we imaged the time-correlated light progression from the outside, and we see that light emission correlates quite well with the expected location to some degree of accuracy. And then when we expand that temporally then, so when we look at earlier light times, so the earliest light times are something like B and C within this image, the early light actually corresponds better to the expected location with respect to the later arriving light, for example, in I and J. And that backs up that the having time resolved imaging ability that we do here with this imaging system is a benefit in providing potentially more accurate location recovery results, which I think was actually quite a decent result. So again, in summary, this is just an initial sort of demonstration of one particular application, in vivo application of this kind of device, but this could potentially be used for a wealth of other applications. But again, what we're seeing is that the early arriving light does potentially, or in this data set at least, it infers a more accurate representation of the actual light source location. So I think that's quite a decent result. Some information about this measurement: we used 100 milliwatts of power, source power, that was from like an isotropic kind of emitting source. So it wasn't extremely high powered or anything like that. We're using picosecond length pulses at around 800 nanometers. Yeah, so some conclusions: just we developed a unique imaging system to optimize photon capture, and that's a standalone kind of device that could be applied for a bunch of different stuff. So again, if you have anything that you want to discuss on that, feel free to ask me. We demonstrated the capability to track the location of an entire fiber device over an extended region. That was in a scattering phantom, which was developed, I did mention previously, was developed to have a scattering coefficient that was reminiscent of biological muscle. And we demonstrated the ability to provide spatial information regarding in vivo light sources within a porcine cadaver model, which I think is the key kind of result of this talk. So we want to expand this kind of work then into further biological models, in this case, an anonymous biological model. And we believe that, you know, based on the results that I presented today, there could be a potential clinical impact from this kind of work in the future. So I don't know what my time is like. I feel like I might be fast, but yeah, I just want to, thank everyone for listening. If you have any questions or anything to follow up offline, feel free to give me a shout or whatever.